Hi everyone, and thank you for joining us today. Welcome to today's Tips and Tricks webinar focusing on preparing EV samples for single molecule resolution imaging hosted by ONI. Before the webinar begins, I'd like to go over some quick house rules for the webinar. Firstly, the webinar will last about 45 to 50 minutes, leaving approximately 10 minutes for Q&A. You are welcome to submit any questions that you have throughout the webinar via the chat section, which is located within your panel. We will be answering your questions during the Q&A session, and if we're unable to get to your questions, then we will email you your answers separately via the email address that you did register with. Please note that today's webinar is being recorded. It will be stored on the media section of our website for future reference. But if you'd like a copy of the webinar as well, please feel free to send us an email and we'll be more than happy to share the webinar recording with you. Lastly, if you're interested in the handout section of your panel, you can find a brochure which is outlining ONI's portfolio and explaining how we can help you throughout your SuperRes journey. Now that we've gone over the house rules, I'd like to introduce you to our guest speakers today, Alison Fujii and Katie Lennon. Alison Fujii is a commercial applications development scientist here at ONI. She has worked in EV biotechnology research, including EVs for cancer therapeutics, stem cell EVs for injury healing, and has worked at ONI on optimizing EV samples for all types of super resolution imaging. Katie Lennon is an applications development scientist here at ONI. Katie recently joined ONI after a postdoc in the Laurent lab studying EV RNA signaling following her PhD in Tiana Talisman's lab, developing methodologies to characterize EVs in human health and disease via super resolution imaging. And now that we have concluded our introductions, I will now hand the presentation over to Katie and Alison to begin the presentation.
Thanks, Caroline. All right, welcome. So today we'll be focusing on EVs for super resolution, in this case, single molecule localization microscopy. And we'll hopefully get you some great images and great data out of the info we're talking about here. So we'll discuss why SMLM is a useful tool for studying EVs. We'll look at the things we need to consider about our EV samples before we begin the imaging process, and what factors we need to optimize during our imaging process after sample prep is done. And we'll be closing with a Q&A. So to start off with, let's talk about SMLM for EVs. A quick review on extracellular vesicles, or EVs, is that they are small membrane-bound particles released by all cells into the surrounding fluids, whether that's biofluids or cell culture media, and they can be uptaken by neighboring cells. And the contents of these cells, DNA, protein, RNA, they can be involved in intercellular communication and therefore can affect the function of recipient cells. Because of this, EVs have a great potential for diagnostics and for therapeutics. So taking a look at this biological size scale here, most of us are quite familiar with using conventional microscopy, such as light microscopes and confocal, to view structures as small as mammalian cells, for example. However, for structures smaller than that, we run into a problem. That's called the diffraction limit of light, which is around 200 nanometers. Below this scale, conventional microscopy can't really resolve these structures with great detail. And unfortunately, EVs fall below the diffraction limit of light because they can be as small as 30 nanometers in diameter. So how can we resolve these? One option is single molecule localization microscopy, or SMLM. Now we can look at things that are nano-sized, such as EVs, viruses, even smaller. So let's take a look here at an example of how we would break that diffraction limit. We're looking here at a fluorescent protein, in this case GFP. It's about 2.5 nanometers in length. However, when we look at this with a fluorescent microscope, we don't see that little structure there. What we see is this blurred fluorescent signal, which is a lot larger than the actual protein, about 200 nanometers in diameter. This is called the point spread function. What we can do with this signal, however, even though it's larger than the actual protein, is we can determine the intensity over the spatial array, and we can look at what the highest intensity is, and we'll fit a point at the center of this point spread function where the intensity of the signal is the highest. And this is where we would localize our protein. So this point then is a lot better resolved than that larger point spread function. Now, what happens if we have some signal like this? What we're looking at here is actually multiple GFP molecules that are close to each other, but how are we going to resolve where they are individually when they're all overlapping? The signals are overlapping. What we then do in SMLM is we will look at just one molecule at a time. So this one molecule is fluorescing independently of the others, and we can localize. Then over time, this molecule begins to fluoresce, and we can localize it. And then later, this molecule is fluorescing and we can localize that one. Now we come up with a much better resolution for where the spatial location of our GFP molecules actually are. So this is important because what we can do then is really resolve EVs looking at things like size, morphology, biomarker distribution. And this is at a smaller than 200 nanometer scale where we can see these details.
But in order to do this, we need to have a good signal to noise ratio. And so the way that you prepare your EV samples is very important for ultimately being able to do single molecule localization microscopy and get images and therefore the information from this type of data. So that brings us to considerations prior to imaging. What do we need to do to prepare our samples in order to make sure that the vesicles that we're imaging are basically real signal and not, background? So first, we're basically going to be talking about multiple different components: EV collection, different purification methods, the capture methodology for how you're going to bring the vesicles onto your surface to actually image, how we do fluorescent labeling, and finally, and probably most importantly, controls. So first, we'll start by talking about EV collection. As we know, and as Alison mentioned, cells release EVs. And so some of the more important components are basically harvesting those EVs under cell culture conditions, focusing mainly on the structure and the function of the EVs in that as we're collecting them, EVs will be taken up by the cells that are in the media. And so it's important to basically be harvesting your media at regular intervals. We prefer to harvest our media every 24 hours over a 72-hour time period. And in that way, you're basically optimizing the most vesicles in your media over time. When you're considering your cell culture conditions, when you're considering your cell culture conditions, you want to be cautious about exogenous EV sources. Most people culture their cells in FBS, and so you don't want to have any bovine EVs in your EV preparations. So you can either use an EV depleted FBS, or you can EV deplete your FBS on your own with an overnight ultracentrifugation. Additionally, you can use media that is serum free. However, you need to be cautious about using, you need to basically choose the media conditions that are going to best suit the outcome for the experiment that you want. So if you're looking at cells in a hypoxic environment that are nutrient deprived, the EV profiles are going to be very different than cells that are in a normal cell culture condition with complete media. As I mentioned, the EV collection, you can essentially collect your media every 24 hours for about 72 hours. We usually do a 300G spin for 10 minutes to remove cell debris. Therefore, you'll be left with a supernatant that contains EVs, and you can keep this in the fridge for about a week. So what I prefer to do is harvest my media at day 1, do a 300G spin, keep that media in the fridge, harvest 24 hours later, do a 300G spin, keep the supernatant, keep it in the fridge, and so on. Once you have all your media collected, you can then do 100 kilodalton concentration, and generally will bring the media volume down to about 0.5 ml. I prefer to do this because of the subsequent purification steps that we'll be talking about later in this presentation. Additionally, you can collect EVs from biofluids. We've collected EVs from blood samples before. Generally, we'll collect the blood in EDTA tubes, which are lavender top tubes. You can store this at 4 degrees, but it just has to be less than 4 hours. Bring it back to your lab, transfer to a 15 ml conical tube, do a hard spin at 1200 G for 15 minutes. This will basically separate your blood into red blood cells on the bottom, a buffy coat in the center, and then your vesicle containing plasma on the top. You can then transfer this into another tube and subsequently do whatever purification methods you're looking at, or aliquot and freeze. Additionally, we've looked at EVs from urine. Again, you can collect urine, store at 4C. Generally, I try to use things as fresh as possible, but you can store it at 4 degrees. Again, transfer to a 15 ml conical tube. We want to do a little bit of a harder spin for urine to remove any bacteria. We're going to do a 2500 G for 10 minutes. And again, transfer into a fresh tube, making sure that you're not transferring the pellet. And then aliquot and freeze, or move on to your EV purification method. We've also isolated EVs from milk. You want to collect your milk and store at 4 degrees. Milk can be stored for up to 24 hours at 4C. Again, transfer to a 15 ml conical tube, do a hard spin. And milk is interesting because at the top, you'll have a fat layer, which you essentially need to break with your pipette tip to get at the EV containing liquid in the center. And so you want to avoid the fat layer, collect the center piece, and avoid the pellet. Transfer to a new tube and either aliquot and freeze, or move on to your purification method. So some of the EV purification methods that people use and that we recommend for SMLM imaging, we're basically going to go through in the next slide. So I know that there's a lot of discussion in the field about all of these different methodologies, but when we're talking about single molecule localization microscopy, we really need very pure EV preparations, and we also need to be thinking about concentration. So my preferred methodology for EV purification is size exclusion chromatography, because you will have a very pure sample. And even though you will end up with a more dilute vesicle population, we do like to do a concentration step following collection of EV fractions. I tend to like this because, again, as I mentioned, the purity is excellent, and also the time and the cost is relatively low. Tangential flow filtration also yields a very high purity, although you can use this to concentrate your samples. You do need to start with a very, very large volume of EVs. It also can be expensive because not everybody has a tangential flow filtration setup, and it can be very time consuming. But generally, when you buy EVs from a company, they are tangential flow filtrated. So you're going to end up with a very pure population of EVs. Differential centrifugation can yield a mixed bag when it comes to purity. It kind of depends on the speed at which you're centrifuging. Additionally, the concentration can be challenging, but in general, I think it's pretty good. Again, the time commitment is relatively low and the cost is relatively low because you can do differential centrifugation on a tabletop centrifuge. Ultracentrifugation, generally when you're spinning at very, very high speeds, you can create aggregation both of your vesicles and of the other particles in your EV preparation. The concentration is going to be very high, but it is very time intensive, and not everybody has access to an ultracentrifuge, so you may have to purchase it or rent it in a core or something along those lines. PEG precipitation, you will probably have some other things that get precipitated, and your concentration will be high, the time commitment is not very significant, and the cost is generally low. But PEG precipitation can also lead to your vesicles fusing together. And so in particular for SMLM, we need to be able to tell where the vesicle borders are, and therefore PEG precipitation can make that complicated. And finally, we have immunoaffinity isolation. This can be with something like magnetic beads, where you're attaching, a PS type of capture or an antibody that you're interested in for a particular vesicle population. You can attach onto magnetic beads, and generally the purity will be good, but the concentration may not be as high, and generally the time commitment is not as significant as some of the other methods, but you will have to purchase reagents for this, and it may kind of add up over time. So in particular, we're going to focus on the two that we use most commonly. Size exclusion chromatography is what I've used for the majority of my career, although when I was beginning my EV projects, I compared size exclusion chromatography with ultracentrifugation and some of the other methodologies as well. And we're also going to be talking about differential centrifugation, which some of the data that we're presenting today was prepared using differential centrifugation. So for size exclusion, as I mentioned, we like to use the ISON QEV columns, and they're great in terms of reproducibility. You'll have an excellent separation between your protein and your EVs. You can collect your EV fractions and pool them together, or you can analyze them separately to look for different size distributions of your populations in the fractions. And generally, as I mentioned, for me, I like to pool them together so that I'm collecting as many vesicles as possible, concentrate them in a 100 kilodalton concentrator down to about 100 microliters. So this ends up being from about 2 mils down to about 100 microliters. Additionally, for differential centrifugation, this is the protocol that one of our customers that Alison recently went and helped them image their EV populations. This is the protocol that they've been using, and it seems to work beautifully. You take your conditioned media, centrifuge at a slow speed to remove any dead cells, and then a slightly higher speed at 2000 G for 30 minutes to remove significant debris, and then a long hard spin at 12,000 G for 90 minutes will result in your EV pellet. And this will generally contain larger EV populations. You may be missing some of the smallest exosomes, but in general, their EV concentration was excellent, and the distribution of sizes was also excellent. So I just want to give you kind of some context with some TEM images about what some of these different methods look like. So again, this is size exclusion. These vesicles are extremely pure, but definitely dilute. Additionally, you can look at ultracentrifuge samples, and you can see that there's a lot more particles in this than the size exclusion, and this will confound your single molecule localization images. And for PEG precipitation, again, we see that it's very concentrated, but we don't have much confidence about what is a vesicle in this type of sample versus what is an aggregate from your PEG or any lipoproteins. And so what does it look like via SMLM? So on the left, we have what size exclusion looks like. This is what we call the good, and ultracentrifugation is not ideal. There's a lot of aggregates, a lot of particles in this. And then finally, the PEG precipitation, we have fusion of some of the vesicle membranes. We have some particles that are extremely round and are probably some sort of aggregate, and basically we just don't have confidence in what is in this sample. So again, we recommend size exclusion or differential centrifugation. And so this brings us to our EV capture and fluorescent labeling. Once you've purified your EVs, how do we prepare this?
Yep. So let's talk a little bit about how we're going to design our experiment before we begin the imaging process. So first of all, let's talk about capture. Why is it important? So what we want to do is immobilize our EV population onto our imaging surface in order to take a snapshot of hundreds to thousands of individual EVs at once. Now, since SMLM utilizes the stitching of many frames over time to create a single image, you can think of it similarly to taking a photo with long exposure. So having captured immobilized EVs on the surface would be good. You want your subject to stay in the focus plane and stay still. What you don't want in a photo shoot is your subject to move in and out of frame, being blurry, leaving the image entirely. So let's talk about the ways we can capture our EVs onto the surface. So there are non-specific capture methods. One example is polylysine coating. So this can be used and it is very sticky. However, because of the stickiness, this may also capture many non-EV materials to the surface, and this can lead to noise and decrease specificity with imaging results. However, you might just want to use it just to see what you have in your sample. Now, moving on to PS-mediated capture. PS is phosphatidylserine, and this takes advantage of the fact that many EV populations have high phosphatidylserine expression on their surfaces. And this method can be very effective for capturing certain EV populations, such as those produced by many commonly used cancer cell lines. But PS is not ubiquitously highly expressed in all EV populations. So it's a case-by-case basis on what capture method you'll want to use. Moving on to antibody capture, there's antibodies such as, for example, anti-tetraspanin antibodies. They can be used to specifically capture EVs onto the imaging surface. And this method can be particularly useful for studying specific types of EVs. Since different EVs may express different surface markers, you can target specific EVs with specific antibodies. Now moving on to fluorescent labeling. How are we going to actually view our EVs? We're going to use fluorescence. So one thing to keep in mind is we recommend conjugated primary antibodies when we're dealing with something as small as an EV. And this is because when we're using a primary secondary antibody labeling system, as you can see in the bottom left image, the fluorophore is actually going to end up a lot farther away from your target epitope, and it can be as far as 30 nanometers away from that target epitope, meaning you're not going to get super great precision with where your signal is and where your target molecule actually is. Additionally, primary secondary systems are a little bit more prone to non-specific binding, and so that can increase background. Now, nucleic acid labeling can be done by using fluorescently labeled oligonucleotide probes that will hybridize to a specific target sequence. You can also use RNA or DNA fluorescent dyes, such as Cyto13 and Yo-Yo1, but there are a slew of different options that you can check out on some of our info pages. Now, we mostly use DStorm when we're imaging our EVs, but there are other methods you can use as well, such as Palm and Paint. So there are potentially different, there are different fluorophores that you might want to use for these different technologies, these different methods. And here's a table you can view here, and definitely go check out more on our website and look up some protocols yourself to design your perfect experiment. All right, let's move on to the actual workflow of how we get from an EV sample to actually seeing it on a Nanoimager. Our first step is surface prep. So here you can see someone pipetting into our EV profiler kit chip a surface prep solution. And on the right, what you see is a schematic of a cover slip and then our capturing molecule. We'll then add our EVs that are then captured onto the surface of the cover slip. And then finally, in order to see them, we will want to stain them with some fluorophore containing markers, or probes. So in this case, we're looking at antibodies that are specific for tetraspanins, as well as biomarker X. And then we will also want to fix our sample. In many cases, we would like to fix it because this helps maintain the actual stability structure of the EVs, keeps the fluorescent molecules exactly in one place. Just like I mentioned before about the long exposure camera snapshot, you like things to be still when you're doing SMLM. And then you would move on to imaging in the next step. Oh, controls first.
Yeah. So when it comes to controls, of course, you need to think about your experimental design and what the goals of your experiment are. So as we've mentioned, generally our fluorescent experiments are using antibodies. And so you need to consider using an isotype control for any antibody that you're using. And especially if it has not yet been published with knockdowns or blocking peptides, you need to make sure that the antibody that you're using is specific for the epitope that you're trying to examine. Additionally, if you're looking at nucleic acids like RNA or DNA, you need to have RNase or DNase treatments where you're essentially removing what should be causing your signal and ensuring that the probe that you're using is specific for the RNA or the DNA that you're attempting to localize. Additionally, we need to have EV detection controls. So you should include a surface or a lane that has no EVs. And additionally, you should also include a surface or a lane that has lysed EVs, just ensuring that the particles that you're localizing are actually membrane vesicles and not some sort of protein aggregate or nucleic acid aggregate. Finally, if you're attempting to look at the content of your vesicles and you're using permeabilization conditions, you also need to include non-permeabilizing controls. And you would expect, of course, in your non-permeabilizing controls that you would not see signal from the content of the vesicles localized with the vesicle membrane. And everyone's experimental design is very different. You may be expecting to see RNA or DNA on the surface of your vesicle, and when you do an RNase or DNase treatment, maybe that signal will go away. And so you need to basically consider your controls very carefully. And if anyone is interested in reading more about this, the MISEV guidelines coming out in 2023 should contain a lot more information regarding important controls specifically for SMLM imaging. Okay, so this brings us to factors after sample prep related to your imaging and analysis. So first, we're going to talk about optimizing the EV imaging and what you need to consider going through that process, and also following imaging, analyzing and interpreting your EV data. So in terms of optimizing EV imaging, what we're really most concerned about is the blinking of the fluorophores. SMLM depends upon the stochastic activation of your fluorophores over the course of your imaging. And the way that we do this is essentially by modulating the buffer to make it a very low or no oxygen containing buffer using enzymes. And so you can read online for different SMLM buffers. ONI, of course, has the one that we sell in our EV Profiler kit. And if you're interested in learning more about that, we can answer any questions related to that. Additionally, when you're doing SMLM, we use what's called total internal reflection fluorescence microscopy, or TIRF. Essentially what that means is that instead of with standard microscopy, the fluorescence is going all the way through your sample and you're getting signal through the entire Z direction of your sample, we bend the light inside of the objective so that you're only exciting the fluorophores at the closest area to where you're imaging. And therefore you reduce background fluorescence from molecules that are higher than the evanescent wave, which is what is activated with total internal reflection fluorescence microscopy. And finally, you need to consider modulating your laser power. So essentially, when we use the imaging buffers that are in a low oxygen state, the fluorophores, instead of just being activated and going back to a dark state, they will be being activated, fluorescing, and going back to the ground state. They will be pushed into this dark state, which you can use the laser power to then bring back to the baseline so they can blink again. So of course, this is all might seem a little bit complicated, but in practice, ONI and others have made it a lot more straightforward to use this for someone who's never considered turf, laser power, or imaging buffers. In our microscope and in our analysis and acquisition softwares, we basically guide you through picking the correct turf angles, choosing the correct laser powers, and if you have any questions related to this, both Allison and I are very happy to answer those. Okay, so now you have your raw data from an EV image. This is what some typical raw data would look like. And of course, thank you very much to Masako Harada's lab, whose data this is. And Allison literally collected this a couple weeks ago. So it's fresh, hot off the press. So you can see some raw images here where you have population of EVs. If we look into this area in the center, we can zoom in here and see individual vesicles with very low background signal. And so what we need to do is essentially use a clustering algorithm to figure out where the border of the vesicle is so that we can then do single vesicle analysis and pull out information like the area, the length, the circularity or the morphology of the vesicles. And then of course, what we're mostly interested in is usually the biomarker content. And so you can see two vesicles here from this sample that are both triple positive and they have very different biodistribution of the biomarkers. And so from this type of data, we can then basically also look at important things, of course, like the size distribution of the entire population. And one of the things that you might notice is that over the span of the diameters, you can see that this is a bimodal distribution. So what's different about the populations of our EVs? Let's take a look at the small EVs versus the large EV populations and see if we can find any differences. So if we look at the small EVs, we have a single positive population, which either has just the biomarker X, CD63 and 81, or CD9. We have a double positive population, which is with biomarker X and the CD63, 81, or biomarker with CD9, or the tetraspanins. And then of course we have a triple positive population, which has all three. If we look at the large EVs, we again see the same populations, but you can see that the number of double positive CD63, CD81 only containing vesicles decrease, and we actually see this increase in the population of triple positive EVs, which contain all of the tetraspanins. And I'm sure a lot of people on this call know that CD9 is expressed generally in microvesicles, which are larger EVs, and so this makes a lot of sense. And again, this is the type of information that you can get with single vesicle analysis, and specifically SMLM is both the size distribution, morphology, and biomarker content of a large EV population. So here's just an example of some of the individual vesicles that were collected from this, and you can see in this top left corner, all of these vesicles are triple positive, and yet they have very different biomarker distributions, morphologies, and sizes. Additionally, on this bottom section here, you can see that these vesicles are double positive, and yet we see very small vesicles that are double positive, large vesicles that are diffusely labeled with these particular biomarkers, and also vesicles that have this ring-like structure that are less densely labeled. And so understanding and preparing your samples in a way that gives you information that we can provide in SMLM imaging can really improve the research and kind of the information that you're pulling, where you're not just understanding the number of triple positive or the number of double positive EVs, but actually where those EVs might be coming from and how they might be functioning in your system. Okay. So I think that this leaves some time for question and answers. Do we have any to start with?
Yes, we do. And thank you so much for the presentation, Allison and Katie. We're now going to open up the floor to questions for our speakers, which they will be answering live. Once again, if you have any questions, please feel free to drop them into the chat box. We've noticed that a lot of attendees are utilizing the raise hand feature in GoToWebinar, but please note that we will not be monitoring this feature. And we'll only be accepting written questions at this time. And like I said, it looks like we have several questions submitted, so let's just dive right into some. For the very first one, we have, when is the best time to store or freeze your EVs?
Okay, so generally I prefer to use my EVs as fresh as possible, but generally in terms of, it depends on the application that you're using. So if we're talking, of course, about SMLM, fresh is best, but if you have to wait, I would leave them in the fridge for a couple of days. If you have to wait longer than a couple of days, then I would go ahead and concentrate them and either purify them and then freeze them, or freeze the concentrated EVs. Do you have a different?
Yeah, I think that that's a good input, because storing things at 4 degrees long term is problematic, but also freeze thaw cycles can be problematic as well. So happy medium, using as many fresh EVs as soon as you collect them would be ideal, but we know that doesn't always happen in laboratory settings.
Right. I guess the only caveat is that for biofluids, it's a little bit different. Generally, I would freeze plasma prior to purifying the vesicles. So, yeah.
Great. All right, for the next question we have, can you plot the biomarker positivity versus EV size for a particular population using SMLM?
Yes, that is something that can be done. So each cluster has a whole series of individual criteria. So it will give you size, circularity, biomarker distribution, of course. So any one of these categories, you can sort it, and then you can plot any category against any other category. So you might see as you get larger, for example, you might see more of a specific biomarker, or you might even see less.
Yeah, you can with the CODI acquisition and CODI analysis software, you can export CSV files. And so then you can pull any information that you want about one cluster. And in that way, you can either use kind of a volcano plot where you have an individual point as one vesicle, or you can do it as bar graphs or representing the whole population.
Right, great. Next question. How long does it take to get results from having purified EVs using your EV profiler workflow?
So the very minimum, if you do everything perfect, is only about 2.5 hours, which could be great because you could finish your experiment in just one day. There are also pause points. So if you'd like to do a longer capture, for example, if your EVs maybe are not quite as concentrated as would be optimal, you can do an overnight capture at 4 degrees. And there are these pause points, so you can take longer and you don't have to do it all in one go and don't get a single break. But so it could be short, or you can break it up.
I think it also depends on how many samples you're attempting to look at. So if you're trying to look at 8 different conditions, both the sample prep and the imaging is going to take longer. So generally, one acquisition with 3 fields of view is about 12 minutes, right? Something.
Yeah.
And so if you're looking at more samples, it takes a little bit longer. Again, because we're separating the imaging over a period of time.
Right, yeah, the imaging, I would say for one single, if you're using our EV profiler kit, one chip has 4 lanes. So if you just round that up, it will take about 1 hour to complete imaging of all 4 samples on that chip.
All right, next question. How do you stain cargo in the lumen of the EV instead of the surface? What if I don't know where my biomarker of interest is located in an EV?
Right. So in this case, we're going to have to get into talking about permeabilization. So starting with not knowing where your marker is, I would say don't permeabilize anything, only stain on the surface, see if you see anything. And then there are different methods you can use to try to permeabilize your vesicles and then image things on the inside. And it's very similar to what you might do in trying to permeabilize a cell and then stain structures inside that cell. Although because EVs are so small, they're pretty fragile. So I think there's a little bit of tweaking and troubleshooting involved in that permeabilization process. And I know Katie has actually been doing some hands-on troubleshooting with permeabilization,
Yeah.
so maybe you have some input as well.
Yeah, definitely. So I think, as Allison mentioned, it's very similar to permeabilizing cells. We have been testing conditions using a bunch of different detergents. I actually published a paper back in 2022 from my PhD lab where we were looking at luminal protein, and we did permeabilization using 0.05% tween, although there's a little bit of differences because it depends on whether or not you're doing your staining in solution or on the capture chip like we're doing now. The one thing that I would just make sure to say is that you need to think very carefully about your controls, especially if you don't know where your particular biomarker is. So if it's on the lumen and you're using a new antibody, you need to make sure that you have an isotype control so that the fluorescence that you're seeing, you can validate that it's truly from your biomarker. And vice versa, if you don't see it and then you permeabilize and you do see it, you need to make sure that you're, again, still maintaining the isotype controls. Also use a lysed EV control to see, is it truly in an intact vesicle? And yeah, so just my advice would be very carefully consider your controls and go from there.
All right, let's go on to the next question. The next question is, the separation of the EV from viruses is challenging because they have similar size and density. Moreover, both EV and viruses obtained from the same cell line will have the same proteins expressed on their surfaces. Do you have any suggestion or approach which could overcome these problems?
Yeah. Have you looked at viruses before?
Not in this context, no. I would imagine that they don't have the exact same surface markers though, although they might share some, which could become a problem when you're talking about capture.
Yeah, I think so. I have not looked at viruses before, but as I'm kind of thinking about it, I would probably try to include a marker for your virus. So like the capsid protein, or if it's an RNA virus, maybe you'll want to incubate, your cells that you're producing these from with something that will label RNA. Yeah, that's a hard question. That's a really, really tough problem, but I would basically attempt to separate the populations based on labeling something specific that's for viruses versus your EV populations. And I think the tip about seeing if there's anything different about the way that you could capture, where you could capture with tetraspanins and hopefully enrich for the EV populations, or you can potentially try and use like a magnetic bead depletion. So if you have a viral protein on the surface, so basically deplete your viral particles prior to depositing your vesicles on the surface, if that makes sense.
Yeah.
Happy also to discuss this, individually, if you want to just brainstorm, always open to kind of talking it through.
Great, thanks so much. Okay, next question. Is there a negative impact on EV production after serum starvation? what time point is appropriate for serum starvation without significantly affecting EV yield?
So I actually have used nutrient poor conditions in order to boost EV secretion before, and it does boost EV secretion. However, during that time, at least in my experience in this particular cell line, they were dying. So in the first 24 hours and then 48 hours, collection of EVs was good, but then starting around 72 hours, the cells were dying, and so they just weren't producing very many EVs because the cells themselves were there were just few of them active at that point. So that's just the timeline that I've experienced before, but I haven't done it with too many different cell lines.
Yeah, and of course it depends on your experimental setup. You were specifically looking at,
Yes.
you were trying to look for stress markers. And so you need to be aware that the vesicles that are released are going to be different than what the vesicles that are released under normal conditions are going to be. So
yeah, that's right.
Yeah.
All right, thank you. So next question, can you image EV inside cells, for example, to assess the ability of EV purified to be taken up by cells?
Yes, I've done this before. It's really cool. They're really, really cool experiments. So you can label, have you done this before?
I haven't, but I'm aware of some of the different methods. You can label them exogenously and then introduce them to cell populations and actually watch it in real time or do time points for uptake. You can also even do experiments where they're labeled inside of the cell, and you can actually see them like being produced and like being released through like the endosomal pathway.
Yep, and you can choose what biomolecules in the vesicles you're most interested in watching being transited. So as we mentioned, I did my postdoc in RNA signaling, and so one of the experiments that I did was attempting to label the RNA and watch it transport from the donor cell into the vesicle population and then ultimately into a recipient cell. So you can specifically label RNAs and then watch them transit, or you can try and label the membrane or label a molecule on the membrane, like a membrane protein or tetraspanin, in order to get images like that. Yep.
Okay. So the next question, how many EVs do you need to get a good sample for SMLM?
That's a really good question. So when we're taking one field of view, it's 30 by 80 nanometers, just what we actually view on the screen at any one time. And what we'd like to aim for is 1,000 or more vesicles in that one field of view. We can still do like data analysis on lower numbers than that, but once you get to around 100 or fewer, that's really not ideal because there's always going to be a certain amount of background noise that is going to be part of your signal or will sort of obscure your signal. So we recommend having multiple hundreds to thousands on that one field of view, and you can also take more fields of view to get more data. And essentially what that translates to in terms of density is 10 to the 9th to 10 to the 11th particles per mil that you're actually applying into your surface.
Yeah, I've done as low as 10 to the 8, but it was really, really challenging. Basically, we had to take a lot of fields of view to get statistically relevant data. So definitely above 10 to the 9, yeah.
Great. All right, next question. Can I think that one fluorescence dot represents one marker fluorescence? Then can you determine the number of markers by the number of fluorescent dots in the image?
Great question. So yeah, so the lab that I did my PhD in specialized in quantitative single molecule localization microscopy, which is essentially equating how many blinks each fluorophore takes to the biomarker that we were looking for. And this is Tiana Talisman's lab at City of Hope. You have to do so many controls in order to do that correctly. it's possible, but essentially every fluorophore and biomarker pair, so antibody attached to a fluorophore or molecular beacon or, whatever you're attempting to look at, has a different degree of labeling. So not every antibody only has one fluorophore attached to it. And not every fluorophore blinks. So basically a fluorophore in the 647 channel may not blink with the same properties as a fluorophore in the 488 channel. So you need to be very careful about equating number of localizations to number of proteins. And I think this is true across a lot of different methods. So as I mentioned, controls here are super important, and you can go and read Tiana's papers if you want more details about how we went about doing that. But essentially we had controls where we had surfaces that were prepared with every biomarker that we tested, and then used the exact probes that we were using in our EV experiments and counted the number of blinks that those fluorophore biomarker combos had, and then basically back calculated how many localizations equated to proteins of interest. So that's a very challenging problem. It's solvable, but it's you have to be very careful and think through your experiment very thoughtfully.
Great, thanks for that, Katie. Okay, the next question. Is the diameter of EVs a good estimate of their size, or do you think they will be flattened on the surface by multiple anchors?
That is a really good question. So first off, we are not looking, when we see EV images on super resolution, we're looking at fluorophores that are attached to specific biomarkers. So already that is a little bit of a proxy for the size, because, for example, maybe a large portion of it does not have a certain biomarker, so you're only seeing half light up. So that's one potential drawback to actually determining true size of the vesicle. And then we are using TURF, so we're only looking at a small slice, and we're generally seeing something that's affixed to a surface like this. Now, because EVs are so small, we're getting a much better size estimate than we would if we were looking at a tiny slice of, say, a cell. But everything is going to be approximate. So you could think of it like a bubble, like blowing bubbles, and it landed on a surface. You're probably going to get mostly a dome shape rather than like a beach ball that lands and only a small part of it touches.
So I actually, I agree that we're basically using a proxy measurement, but I have cross-validated single molecule localization images with TEM and also, nano-sized and other like size estimation methodologies, and they all correspond. So we are looking for morphological differences, which we can tell, and we are looking at size distributions, which if you're looking at the population of vesicles, you are going to have quantitative data about the size distribution compared to each other. And that corresponds with what I was seeing in TEM, and it corresponds with what we were seeing with nano-sized as well. So yes, this is a potential limitation, but it's not significant in my opinion and in my experience. Yeah.
Great, thanks both for that. All right, the next question is first thanking you both for the great talk, and then saying, have you tested any nucleic acid markers and how they look with this imaging technique?
I have, yes. So in my PhD, we were attempting to look at RNAs using molecular beacons, which are essentially hairpin structures that have a quencher and then a fluorophore, such that when the molecular beacon binds to a microRNA, it will open up and you'll have fluorescence from your fluorescent marker. Those experiments are challenging, and you have to basically design your molecular beacon for the particular microRNA that you're attempting to examine. In my postdoc, I looked for non-specific RNA labels. So one of the things that we were attempting to do was to incorporate non-natural amino acids into the RNA, for example, using 5-EU, and then using click chemistry and permeabilization conditions to basically click on an Alexa Fluor dye directly to the RNA. And yes, I was able to essentially quantify the percentage of the vesicle population that I was seeing that was RNA containing, which was awesome. It was really fun. And that was actually the first time I ever used a Nanoimager, because I had left my PhD lab, which had an amazing microscope, amazing setup, and analysis software kind of built in-house. And I was in a postdoc that we didn't have someone to analyze my images for me, and we didn't have access to a super res microscope, and I found a Nanoimager in our building, and it was a really cool experiment. So that was the first time I had used a Nanoimager. Yeah.
All right, wonderful. So we did receive a pretty overwhelming number of questions today, but we only have time for a few more. So moving right on to the next one. Which fixation do you use due to fixation-sensitive dyes?
Yeah, so what my experience has been so far is generally we use PFA and avoid dyes that are fixation sensitive. Or you can also skip fixing. It's maybe won't give you the most precise resolution, but if you are set on your specific, maybe you're using a tandem dye or a certain hydrophobic dye that you really want to use, yeah. I think that there are some resources that we can provide you about the best matches between either like certain fixatives and then certain dyes, or maybe ways to get around the fixation step.
Great, okay. Next question is, oh, my question screen just went away. Just give me 1 second.
I guess we
All
can.
right, sorry about that. We are wondering if there are any adjustments that need to be made to the EV kit prep and imaging protocol when working with EVs isolated from different species. And they put as examples, human, horse, mouse, cattle.
Yeah. So my understanding right now is that the capture antibodies that we use are tetraspanins, anti-human tetraspanins generated in a mouse. So essentially, if you are looking for a species-specific marker, you need to ensure that that the antibody that you're using to capture can recognize the epitope from that species. So essentially, yes, you need to think about this and consider it in terms of what you're going to isolate. Yeah, I think that's essentially.
And just notably, our profiler kit is modular, so whatever marker you want to use for your specific EV sample, you can just sub that in for the provided, capture, the capture antibodies or the capture methods that we include.
And I think in general, our support, we want you to be successful in your experiments, whatever you're attempting to look at. And so feel free to reach out to us and, as a company, and we'll attempt to help you solve your issues.
Okay, probably gonna let 2 more questions come in. The next question is, how about biotinylated primaries? And it was noted that this question was asked during the EV capture slide.
Okay, so are you, I'm wondering about a little bit of clarification here. So biotinylated primaries for capturing, that's absolutely a great way to do it, because that's actually the scaffold upon we build our surface prep. So if you have a primary antibody that is specific for a certain marker that's on the surface of your EVs that you want to use to capture, yeah, flow that in. that works perfectly.
It'll attach to the surface
Mhm.
and then you can put your EVs in and it'll capture.
Yep. If you're talking about staining, then that's also a possibility too. If you have a biotinylated and then you have some probe that is, streptavidin, you can do sort of that building a scaffold. Then you also might want to consider, having your fluorophore being a little bit farther away from your actual target, but it's, give and take for your goals in that experiment.
Okay, and I'm going to feed you guys one more question. Does the storm buffer require a small amount of oxygen presence, or we should try to achieve a completely oxygen-free environment?
So our storm buffer it's not gonna ever be completely non-oxygenated. I would say do your best, don't vortex it, don't pipette a lot of bubbles in there, but we have an oxygen scavenger molecule, an enzyme that's in there, so it should be cleaning up like excess O2 that is present. It's always going to be there to some extent, and just, best techniques to keep it clean.
And it's also dependent on your fluorophores. So for example, Alexafluor 647 prefers to have as little oxygen as you can possibly get, like as close to 0% oxygen as you can get. But some of the fluorophores in the other channels actually blink best at about 2% oxygen. So if you're doing a multicolor experiment, you want to be kind of in the balance of that amount. And of course, like air has 5%. So our normal buffers have about 5%. So we're basically bringing that oxygen level down to achieve the best blinking in as many channels as you're interested in looking at. So that's basically the goal of our imaging buffers that we sell in the kit. And also if you're considering other imaging buffers, make sure that you're looking at all of the channels that you're attempting to image in and optimize from there.
Okay, it seems as if we're now at time. If we were unable to get to your questions, we're going to do our absolute best to contact you and answer your questions individually. I know several more came in, so we thank you for that. And once again, thank you so much for the presentation, Katie and Allison, and thank you to all the attendees for joining us today. Upon exiting the webinar, you will receive a survey. Please fill out that survey to provide us feedback on your experience today. And if you're in need of being contacted directly by ONI for more information or updates on our offerings. Thank you again, and we hope that everyone has a wonderful day.